Hello students. So, in this video let us be discuss regarding one more important concept, that is genetic code, right. So, in previous video we discussed about the transcription, right. So, transcription and translation both are required, right. These are the two important steps in protein synthesis, right. So, but before going to start the translation process, so we need to understand that is genetic code, because the what you call the synthesis of protein, right, synthesis of protein, it is a polypeptide chain. Protein is nothing but it is a polymer, right, it is a polymer in which there are number of monomers, they are called polypeptides, polypeptide chain, polypeptide strand, right. Protein is nothing but that polypeptide chain, right. So, this is formed by the mRNA with the help of what you call the different tRNA, rRNA, and in presence of amino acids in the cytoplasm of cell, it will be conducted, usually what we call in the ribosomes. So, hence we call it as ribosomes are the protein factories, because the protein is synthesized in the ribosome, right. So, but it is a complex process where the protein is synthesized. Once again we should recall, so that one of the important what you call central dogma, right. So, that is important for this understanding, that is central dogma, in which central dogma. So, this explains us regarding the how protein is synthesized. So, here the DNA, how it forms the mRNA, and mRNA how it forms the protein, right. So, here DNA can produce the mRNA, that is called transcription, right. So, and mRNA forms the protein, that is called translation, is it right, translation. So, here we need to understand the genetic code in this case, that is the how mRNA converts that what you call amino acids into a protein. So, how it binds, right. So, and which amino acid should join one after one, that is in the sequence of codes, right. So, actually DNA, so we actually the genetic code was proposed by George Gamow. His name is George Gamow, right. So, the name of the person who discovered genetic code or proposed it, so that is the George Gamow, right. George Gamow proposed the RNA after processing hnRNA into mRNA. So, that is the mRNA is now formed, right. So, this mRNA how it forms the protein. So, let us we take the one point here, the process of translation, that process of translation, this process what do you call process of translation is the transfer of genetic information, very important, genetic information from a polymer of nucleotides, right, a polymer of nucleotides to a polymer of amino acid, to polymer of amino acid. So, polymer of amino acid is nothing but protein, it is nothing but protein, right. So, polymer of nucleotide is nothing but it is the genetic material, So, whatever the information which is present in the genetic material, is it right, genetic information, that is said to be genetic information. So, this genetic information can convert into protein, right. So, as I explained earlier, so this is what the nucleus, and this may be a cell you can consider. So, here there is a huge amount of what you call DNA is there. So, that is double stranded, as you know, DNA, right. So, this DNA contains the sequence of DNA, no, it is DNA, A, T and guanine, cytosine, right. These are the nucleotides, right, nucleotides, adenine, thymine, guanine, cytosine, right. adenine, thymine, guanine, cytosine. Adenine, guanine are parents, pyrimids, right. So, no doubt 4 nucleotides are there. So, these 4 nucleotides are like what we observed in computer language. So, whatever you are giving data, so that the computer will take it in either 0 or 1, 0 or 1, right. So, in that combination it will take. So, here also the combination of these letters, these 4 letters, right. So, there are 4 letters here A, T, G, C. So, but what it is? So, on mRNA is prepared already because that is complementary, right. So, mRNA is complementary to the subscribing strand of the DNA. Yes, already we discussed. So, for example, this is the DNA. So, this is the DNA, one is coding strand, another one is non-coding strand, or what we call a subscribing strand. To the subscribing strand it is complementary, and to the coding strand it is similar, only the base pair is instead of T here it will be U, AUGC. So, it is become now yes RNA, right. This is the RNA. Is it clear? So, on RNA there are AUGC, these are the 4 letters. So, in this combination, so when you make this combination and permutation, so there is actually 64. So, there is a formation of 64 codons, right. So, let us we discuss it later. So, after what is codon and what is anticodon, let us we discuss again once again go through this one. So, first point I think clear it is a process of translation. This is what the translation process, mRNA to protein, mRNA to protein. So, the process of translation is transfer of genetic information. So, what it is actually, the mRNA is not converted into protein, right. mRNA directly not converted into a protein. So, where mRNA is involved in the protein synthesis, how? In the form of codes and anticodes, anticodons, what we called. So, that is what that is transferring the genetic information in this format. In which format? So, that is in this form AUGC. There are codons nucleotides are there, no. So, these nucleotides are arranged in a particular pattern, and there is a formation of sequence. As per that sequence, the 3 consequent codes we can say, so or nucleotides or base pairs. So, we can use the term base pairs or nucleotides. When the 3 means continuous 3, that is also called consecutive 3 set of nucleotides are referred as triplet, that is called codon. Triplet codon, here what I have taken triplet. So, usually genetic code that is simply it is a signal or a code, we can say it is a signal or code system, it is a coding system, it is said to be a coding system in DNA, coding system present in DNA to control the synthesis of mRNA and protein. Both the synthesis of mRNA and protein is controlled with the help of a signal system, that is what we call a coding system, which is existed in the DNA. Already DNA are having, yes, DNA is nothing but, so there is a what is there? So, nitrogen base pairs, the arrangement of base pairs, that is in the form of nucleotides. There are 4 types of nucleotides, based on this arrangement. So, the code we referred this here, whatever genetic code we are calling, that is in the form of codon. The special term we are giving for this code, codon, right. Codon is in the form of triplet. Why we call it triplet? Because the codon is a set of, it's a set of 3 consecutive nucleotides. 3 consecutive means neighboring, continuous. For example, this is what the mRNA here 1 2 3, this is what one codon, and next 1 2 3, this one, right, in this form. So, it is a set of 3 nucleotides or 3 base pairs, right. So, out of this either this 3 or either this 3, so or the combination, when you make the combination of and permutation, So how much number we will get? So about 64. 4 numbers are here, 4 letters are there. The combination of these 4 letters will become 64. That means 4 into 4 into 4, right? 4 4s are 16, 16 4s are 64, right? So this much of codons we will get. Okay, out of that 3 are not codes for any amino acids, hence we call it as a anti-sense codon, or also called a nonsense codon, right? We also refer it as nonsense codon, right? And 61 are said to be which are coding for amino acid, so that is codons. We are called it as codons, and out of that 64, 3 are remaining, so that 3 are said to be nonsense codon because they does not code for any amino acid, right? So understood now? I hope this is the important point here. Codon is a set of 3 consecutive nucleotides on the mRNA. mRNA means AUG, AGC or AUC or UGC or GCU, whatever. Every combination we will get here. Okay. So later on we will see that the combination of that 4 nucleotides, right. So understood now? Codon is a set of 3 consecutive nucleotides on mRNA, right. So this is clear. So there are many codons. There are many codons on mRNA. What I have written here, there are many codons. For example, this is one codon, second codon, next third codon, fourth codon. In this way, there are many codons, right. So if it is third end, this is up to fifth end, right. So in this way, on this along the length, so there are many codons on mRNA where each codon, for example, this first codon codes for an amino acid. This one codes for a amino acid. For example, that amino acid is, you consider it as amino acid 1, right, maybe methionine or lysine, right. So any one, usually first one is usually AUG will be there, that is the initiator codon. After studying the silent features of this genetic code, we will understand that really. Okay, in the next video we will see. So understood now? According to this line, there are many codons, right, 1, 2, 3, 4, like triplets are there. So that are many triplet codons present on the mRNA, where each codon codes for an amino acid. Each codon codes for an amino acid, right. So next one, the based on the codes present on mRNA, based on this, the codes which are present on the mRNA, this is what the mRNA, right, this mRNA, based on this, there are a set of 3 nucleotides on tRNA also. Based on this, a set of 3 nucleotides on tRNA, especially tRNA diagram you know. Okay, we also call it as anti-codon loop, because that 3 set of codons are called anti-codons, right. So this is what the 3 set what we referred. So it was this is what we call anti-codon loop. We called it as anti-codon loop. There are 3 consecutive, a set of 3 consecutive nucleotides present on tRNA are called anticodons. This anticodons it reads the mRNA. Which anticodon should come here? If it is AUG. So for this it is become complementary. Yes. So AUG will brings always methionine, the first this one. So against to this one there is a usually what you call, there is no complementarity also. Okay, there is no complementarity. AUG is the codon, and for that anticodon will be there here. So that anticodon will bring one amino acid, that will called as amino acid 1 here, right. So that's what, based on codes present on mRNA, based on the codes present on mRNA, there are set of 3 nucleotides, on tRNA, so are called anticodons, right. This is what the codons and anticodons you should remember. yes, and there are totally 46 triplets are there, triplets. Out of this 61s are referred as codons because they code for the amino acids, right. So, only there are actually 20 amino acids, right. So, even this 61 codons are code for 20 amino acids, right. So, more than one codon can code the this out of 20, right. So, but it is said to be universal. We will discuss that in the silent features. So, related to that one, you remember at least this one here in the introduction part. So, it is the introduction part what I am giving here. So, you remember for the genetic code this one, this point you should remember. Genetic code is a signal or a code system in DNA to control the synthesis of mRNA and proteins. And here if the codons changed or nucleotides, consecutive nucleotides change in the genetic material, what you call mutation. So, the change in the genetic material leads to a change in the synthesis of protein, change in the sequence of this one, change in the sequence on mRNA that could change the synthesis of a kind of protein, right, because instead of one particular amino acid on third code. So, if it is changed here, if it is changed, okay, so one more it will become different code, okay. So, it can join, it can bring the different amino acid and it will place there. So, by that the protein property will be changed. So, it may become different protein, right, the same protein may not be synthesized there. So, change in the genetic material cause change in the synthesis of protein, that also you should remember, right. So, these are the points you should write in the introduction part of genetic code. So, later on we will see the genetic 64 triplets are there, no, with the help of a simple the dictionary, right. So, with 20 amino acids, what are those 20 amino acids and what are those 64 codons, we can make that chart in the next video. Thank you, students.